नीचे बैठने के लिए ज्यादा मजा आ रहा है। These two girls come here.
एक साथ बोले मैम प्रॉब्लम है।
बड़ा सबका इन लोगों का प्रॉब्लम तुमने समझ के रखा है।
मैम मुझे भी ठंडी लग रही है पर आप एसी बंद करने नहीं दे रहे।
नहीं एसी मत बंद।
आराम से बैठो।
मैम फैन बंद।
मैं तो सब हाँ
क्यों? 15 मिनट मेरा एग्जाम जेनेटिक स्टार्ट किया
15 मिनट में।
अभी आज पहले तो ये चैप्टर कंप्लीट कर दे। चलो ना नो डिस्टर्बेंस दैट साइड। श्रीराम और तन्मय आज एक साथ बैठे हैं तो आई हैव टू स्पेंड हाफ एन आवर डिसिप्लिन में। तुम पढ़ रहे हो तो दिख रहा है तुम्हारे मार्क्स से।
पढ़ रहे हैं।
चलो we are going to complete with this chapter on photosynthesis. No, we are studying photosynthesis.
हम लोग पहले ही एक साथ बैठे चल ही रहा है।
कल
हां मैम कल हम लोग एक साथ बैठे।
हाँ तो फर्स्ट में बैठे थे ना। फर्स्ट में बैठना और लास्ट बेंच में बैठने में फर्क है ना। I gave you some questions to be done as homework no yesterday. So yesterday we completed with like external factors we were doing. Finish.
Yes.
You were not there only in the class.
I was there.
तो ये चैप्टर
Physically तुम थे. I have only finished with carbon dioxide and light intensity. Yes, Prisha? Did I finish with all the external factors? Not at all.
We have done internal factors that
अरे वाह.
external factors भी क्या है? Temperature
इस तन्मय से मुझे ये expectation नहीं
I'm
था.
sorry ma'am, I'm temperature का.
Now, we are going to do that, and on top of that, we are going to also start with the experiments. अच्छा, now first of all, रखो इधर रखो. First of all, complete this. Take out your textbooks.
Ma'am, please.
Page number. Don't give it to me. You tore it. Now you are going to give it to him.
I didn't do that.
Now, page number 78. B sub number 1. Karo. B sub number 1. B sub number 1. Number 2. Number 2. Number C खाली नहीं चाहिए. A, B, D and E you can do. A, B, D and E. A, B, D and E you will do. Number 2, fill in the blanks you will do. करो फटाफट complete this one chapter fast.
A, B.
I'll repeat. I said number 1, B का number 1 करना है, number 2 करना है, excluding C. 2C अभी छोड़ दो. Leave out 2C and then go to C. Short answer type C. Number 2. Number 1 बाद में करेंगे. We are yet to finish some parts of the chapter. C number 2. Let me see if you all are able to answer all these questions. Complete करो fast. B का 1 and fast. Just 5 minutes. Complete करो. You were not there, no, last class. Why? You were online but yesterday. चलो complete करो fast. I'm going to ask you the answer just in another 3-4 minutes. फटाफट करो. Source of CO2 for aquatic plants. Source of CO2 for aquatic plants would be respiration of aquatic animals. No? What is the option given there? Name the following. Source of carbon dioxide for aquatic plants. What can you write now? B का first and second करना है, C का second करना है. Animal respiration only, there you have to write. Animal respiration. Aquatic animal respires and they release carbon dioxide which remains dissolved in the water. No. So there you will have to write animal respiration. Source of carbon dioxide for aquatic plants. Animal respiration. Animal respiration. Animal respiration. No need of writing fishes because there are other aquatic organisms also. Many. So in general, you write animal respiration. ये सही है ना कि हर जगह ओपन हो रहा है हमारे यहाँ पे थोड़ा क्राउडिंग हो रहा है चेक करके रखो चलो फास्ट डन डन different book. If you have the other book, this side, you may start solving the name the following and fill in the blanks from your book itself. It will be a similar set of questions only. नहीं है? What type of questions are there? अच्छा सॉल्व कर लिया है ओके गुड What's her name? हाँ चार्वी हाँ बोलो the first question number 1 Question number 1, what I gave you? Name the following. Oh, B, yes. Name the following. Start, answer the first 3. Answer the first 3. Could you complete? Okay, na? Chalo, complete karo. The first is all of you check your answers. If you have written it correct, good. The category of organisms that prepare their own food. Autotrophs. Okay, that part we have to do it. The kind of plastids found in mesophyll cells. Kind of plastids. Chloroplast. Chloroplast. Kind of plastids found in mesophyll cells. The compound which stores energy in the cell, ATP. Next, Tanmay. The tall one. Glucose. Next, already we discussed, no, the source of carbon dioxide, animal respiration. Next is the part of chloroplast where the dark reaction takes place. Stroma. Okay. So all of you got this. Number 2. We move on to number 2. You have to write the reason, the reasonable word there, the logical word there. Chlorophyll, magnesia, hemoglobin, iron. Hemoglobin, iron. Which type of mineral element they are talking about? That's a logical sequence there. Light reaction, granum, dark reaction, stroma. Respiration, carbon dioxide, photosynthesis. Producer, I told you we have not finished that part. So producer, autotrophs, consumer, heterotrophs. Okay. Respiration, carbon dioxide, photosynthesis. Respiration, carbon dioxide, photosynthesis. I'm sorry.
That is a product.
Respiration, carbon dioxide.
Oxygen is the prime.
Photosynthesis. Oxygen. क्या लॉजिक क्या है? तुमने बोला था और क्यों लॉजिक?
क्योंकि कार्बन डाइऑक्साइड रिलीज होता है रेस्पिरेशन में और फोटोसिंथेसिस में
ऑक्सीजन रिलीज होता है. So that's the logical concept that we have to use here. That here they are talking about what is getting released as a result of that process.
Now, what is the primary product of respiration?
Primary product.
Respiration. After respiration, what is the
Primary product. Okay, but no primary product है तो तुम बोलोगे photosynthesis में oxygen is not the primary product. नहीं. यहाँ पे if they are talking about respiration, carbon dioxide along with carbon dioxide, large amount of energy that is also a primary product of respiration. So यहाँ पे we are not talking about glucose, carbon dioxide है तो oxygen. Product, main product, byproduct पे नहीं. The type of the gaseous component कौन सा? That way. Last one, water and minerals, xylem, prepared food, fluoride. Okay, move on to number 2. Finished all of you? Yes. Finished all of you done? That's it. अच्छा तुम लोग का बुक अलग है तीनों का. Selena, you did not get it? You don't have the book right now? Achha, it's okay. Did you do? Complete ho gaya, bolo. Number 2. Fill in the blanks. Number 1, bolo. Site of light reaction is? Grana. Next. The chemical substance used to test the presence. Achha, this we are yet to do it. You may leave it for the time. Huh? Achha, if you know it, very good. Otherwise, I'll ask you only when we are done with that topic. Number C. Stroma is the ground substance in chloroplast. Number D. The dark reaction of photosynthesis is also known as Kelvin cycle. In the flowering plants, food is transported in the form of sucrose. Food is transported in the form of sucrose. Soluble. Huh?
I mean, the food is made in glucose, transported in starch, and then stored in starch.
Correct. The food is formed as glucose, but immediately it gets converted into a little bit complex form, sucrose, and then stored into even more complex form, starch. Okay. So this part of the question and the rest of the question we'll do after we finish the chapter. So now next is external factors we haven't finished. So let us complete with external factors. And external factors like the light intensity and carbon dioxide we're already finished. And now we have to talk about temperature. What is the relation of temperature with the rate of photosynthesis? Why are you holding his hand? तुम लोग का ये सब बिहेवियर देख के ना डर लगता है। एक तो को कभी कभार आता है और आता भी जब है तो ऐसे ऐसे हरकत करता है।
This was not expected.
I know. Now there. इसको डाउट है।
इसको डाउट है। मुझे डाउट नहीं है ना। हम कंटिन्यू।
कितने परसेंटेज से पास कर तो पहले वो सोचो।
अभी वो पूछ रहा है।
25 साल में शादी करेगा वो।
उसका बच्चे की भी बात कर रहा है।
मैं नहीं रहा दुर्वेश पूछ रहा है।
ये हेड बॉय।
मैं पूछ रहा बच्चे कैसे डॉक्टर करे।
रास्ते के बीच में ले जाके मारूंगी पता है। Funny creatures you all are. Now, when we are talking about this, all of you look here.
Yes, ma'am.
Shri Ram.
Yes, ma'am.
Temperature, what is the relation of temperature with the rate of photosynthesis? So now we are talking about the rate of photosynthesis and we talk about temperature as an external factor. One concept already you have studied that a particular temperature only can allow the activation of chlorophyll. I already told this, that whenever we are talking about the initiation of the process, what is the first step? Activation of chlorophyll. Activation of chlorophyll will happen only at a definite light intensity and also temperature. So I made it very clear that if I talk about that at the start of the day or daytime में light reaction होता है, जरूरी नहीं है. The daytime meaning any part of the day the process of light reaction may happen. I may also understand when it is a very gloomy and monsoon day, monsoon time, you may understand by the time it is 11 o'clock also, if it is not optimum temperature and intensity of sunlight is not optimum, maybe at 11 o'clock by the daytime the process of photosynthesis may not start. So it is not only that the start of the day and the start of the process. It has to be a definite amount of intensity of light and a optimum temperature. तो सबसे पहले क्यों चाहिए optimum temperature? Activation of chlorophyll के लिए. That's the most important thing to understand and remember. So now when we are talking about temperature, yes, at the start of the day, we understand that the temperature is optimal. The temperature is suitable for the process to start and the rate of the process keeps on happening. But after a particular rate, after a particular rate of increase of the process, when will the process almost come to a stable, or when, first of all, now that I understand that the process is rising up or the process of photosynthesis just starting, and when will the process take up a spike when the temperature is optimal? So I may understand that the temperature may be something around 18 degrees, or I may understand that the temperature is something around 20 degrees. But when the temperature is something around 34, 35 degrees, the rate of the process will be very, very high, because something around 34, 35 degrees, that is what is considered to be the optimum temperature, and the rise of the process will be very high as long as that temperature is maintained. But here temperature will not be maintained for a greater part of the day. It's not so. That temperature will be maintained only for a particular duration, maybe for an hour or half an hour, 45 minutes, whatever. So at that part of the day, the rate of the process will be the highest. But again, if the temperature continues, and if the temperature continues, maybe now the temperature has reached something around 40 degrees. So what will happen? Initially, there was a big rise, a spike was there. Initially, a big rise was there. But now that the temperature is increasing, 37 ORAC, 38 ORAC, what will happen? Is the rate increasing sharply? No, it is not that increasing sharply. Because 34, 35, something around 35 and a half तक तो बहुत अच्छी तरह से जा रहा था. But now it is more or less on the declining side. Now it is more or less on the declining side. Why it is more or less on the declining side? Because the temperature has increased quite a lot now. So here, दो चीज याद रखना है. When the temperature is something around 34, 35 degrees, 35 degrees considered to be the most optimum temperature where the process of photosynthesis will happen at the highest rate. So whatever be the duration, ये temperature जब तक maintained रहेगा, the process will rise at a alarming rate. The process will go on very smoothly, very nice. But as the temperature increases, the process is still going on. But if the temperature gradually increases and reaches the higher, touches the higher level, something around 39, 40, are we to understand that the rate is still increasing and increasing? No, it is just the reverse. So I would say relation of temperature and the rate of photosynthesis till one particular temperature, it is directly proportional. Temperature भी increase हो रहा है, the rate is also increasing. But after a particular temperature, it is inversely proportional. Temperature increase होता जा रहा है, but the rate will come down. The process is almost slowing down, and something around 40, 42 तक तो the process will stop. Something around 40 and above, the process will stop. Now, why will the process stop? Why after 40 degree, the temperature, when the temperature rises something around 40 degree, why does the process stop? The process stops because most of the enzymes which are necessary for the process of photosynthesis are proteins, and they start breaking down because of that high temperature. That is what is called denaturation. That is what is called denaturation of the enzymes. Denaturation. What is the meaning of denaturation of the enzymes? Breaking down of the enzymes. What is meaning of breaking down? I would say that the enzymes are nothing but some protein chains, and because of the high temperature, they may start getting broken down. The bonds are breaking. The enzyme has lost its efficiency. It cannot carry out any function. So, अब enzymes ही functional नहीं है, how will the rate happen? So, ये है one concept to understand what is the relation of temperature with the rate of the process. एक और concept ये है कि whenever I'm talking about the rate of the process and temperature, इसमें quite a number of times this also has been understood that initially, say suppose I understand that the temperature is something around 15 degrees. And now the temperature is something around 15 degrees and the rate of the process is steadily increasing. Now suddenly, maybe now from 7 o'clock till 8 o'clock, the temperature was something around 15 degrees and 16 degrees and 17 degrees, gradually increasing. And the rate is also almost flat, but it is increasing. But suddenly now we get to see that in just next 3 minutes, the temperature has just increased by 10 degrees. The temperature has just increased by approximately 10 degrees. Then what will happen? ये जो धीरे-धीरे increase हो रहा था, now it will take a sharp rise. ये जो धीरे-धीरे increase हो रहा था, now it will take a sharp rise, and again it may become stable after some time. Again, it may become stable after some time. But again, if the temperature rises, and maybe now it is something around 30 degrees, or again, I may say that something around 29 degrees. And after 29 degrees, if there is a sharp rise by 10 degrees, अब वापस it may show a rise. But now that after 39 degrees, it is approaching 40 degrees, अभी और rise नहीं होगा, it will show a decline. So what are you understanding from this concept? We are understanding that काफी बार the temperature remains stable for a particular period of time, like something around 2-3 hours. The temperature may be something around 22 degrees. The rate of process is also stable. The rate of photosynthesis is also stable. But something like maybe by 12 o'clock, suddenly the temperature has increased from 22 to 32, a big rise. You see nowadays, no, what's happening nowadays if you have analyzed. Maybe in the morning by 9 o'clock, it is still glooming. But by 10 o'clock, 11 o'clock, there is bright sunshine again suddenly. No? So there's a sharp rise in the temperature. You will see that suddenly it has become so hot and the temperature is increased. Again, by 3 o'clock, the temperature dips down. Again, it becomes a little bit gloomy. So yes, this happens. So whenever I'm saying that suddenly when there's a rise in the temperature by 10 degrees, suddenly there will be a higher rise in the higher spike in the process. Suddenly when there will be a rise by 10 degrees, there will be a higher rise in the process. But if it is something around 40 degrees now, it will stop. So three things here to understand. At an optimum temperature, the rate of the process works the best. But which temperature it is? 35. So 35 degrees centigrade plus minus 2-3 temperature we can consider. But for your answer, you are going to write 35. So 35 degrees centigrade is considered to be the optimum temperature when the rate of the process is the highest. Then, if the temperature is steady for a longer period of time, what will happen to the rate? That also will remain steady. Then, is there any chance of a sharp rise? Yes, if the temperature rises not by 2 degrees or 3 degrees, but by 10 degrees almost. If the temperature rises by 10 degrees, even in cold countries, what happens? When the temperature in the morning may be something around 2 degrees, 4 degrees. But maybe by 9 o'clock in the morning or 10 o'clock in the morning, suddenly there may be a rise something from 2 degrees to almost 12 degrees. In cold countries, that happens. So now, suddenly by a rise 10 degrees, then the rate of the process has increased. But anyways, whatever it is, if it is rising up also from 40 to 50, if I understand the Middle East, countries in the Middle East, when the temperature rises from 40 to 50, yes, it happens. So are we understanding that the rate of the process also will jump that high? No, it will stop. Anywhere you talk about, it will stop. And the reason already I told you. So that is about the role of temperature. What is the I said reason is why at a very high temperature the rate of the process stops abruptly because of the denaturation of the enzymes. The enzymes all start breaking down. Enzymes break down, that's why. So this is about the enzymes and the temperature. Now we are going to talk about internal factors. Now internal factors, basically two internal factors you are going to remember. Denaturation of enzymes in the chloroplast, because chloroplast is the site where photosynthesis is happening. Now, what are some internal factors? What are some internal factors that will regulate the process? Internal factors, एक है chlorophyll. Internal factors, एक है water content. Water content. So, whenever we are talking about the internal factors, सबसे important है chlorophyll and water content. Now, when we are talking about chlorophyll, already we understand. Chlorophyll and the rate of the process, क्या link होगा? Chlorophyll के साथ rate of the process directly or inversely proportional होना चाहिए? Directly. Adequate the amount of chlorophyll, higher will be the rate of the process. Is it possible ever that a plant cell or a plant is not able to produce adequate amount of chlorophyll? No, ho sakta hai, because there are many such conditions where the leaf cells may not be exposed to adequate amount of sunlight, chlorophyll production adequate nahi hoga, or there are certain leaves where the amount of chloroplasts may be much lesser. For some reason, the leaves may not get adequate amount of water. For some reason, the leaves may not get adequate amount of water. Now, how can water generally allow the process of photosynthesis to run?
Now if water is content, then the turbidity and stoma.
Okay, that is one. So if I'm talking about water content, first of all, we have to remember the stoma, the opening of the stoma, which should the stoma should remain open for a longer part of the day so that the carbon dioxide diffusion happens. We studied yesterday. Or kya? Protoplasm, the fluidity or the semi-fluid nature of the protoplasm is very much necessary. When I'm talking about the water content, if you remember, at the start of this chapter only, I said process of photosynthesis happens at the surplus of water. What is the meaning of that? For the process to run, the matrix or the cytoplasmic region of the cells, mesophyll cells, chloroplast, जो ground substance है, cytoplasm है, which has got a large number of enzymes and pigments, they should have adequate amount of water to be in the functional state. अब जो water नहीं रहेगा, the protoplasm will start drying out, no? If the leaf cells have deficiency of water, protoplasm will start drying out. So in that dried out condition of the protoplasm, do you think all the chemical reactions will happen adequately and efficiently? No. So that's the reason we say that water also plays an important role when we are talking about the process, the rate of the process. So ये हो गया internal and external factors which regulates the process. Yesterday we had also studied about the adaptations. Now we move into another important section of the chapter जिसपे एक question हमेशा that will be there in your paper, diagram based questions on photosynthesis. Now for that we are going to understand first of all a concept known as destarching. What is known as destarching? What are the steps and what are the reasons? Reasons also you are supposed to understand. But first of all, if I understand destarching मतलब destarching से क्या समझ में आ रहा है? What is destarching? Starch को remove करना. So first of all, if I understand destarching, what are the steps of destarching? The first step, we select any particular plant, collect some few leaves, and then what do we do? The first step is boil the leaf in water. Boil the leaf in water. Why? What is the necessity of boiling the leaf in water? Kill the germs. Kill the germs. Kill the germs, you think? Kill the cells. Kill the cells. Not to write kill the germs at all. Mostly students would write kill the germs. Because boiling ज्यादातर हम germs के साथ ही attach करते हैं ना. But यहाँ पे we are not talking about killing the germs at all. We have nothing to do with the germs here. We are talking about why should the plant cells, the leaf cells should be treated with hot water or boil it for 2-3 minutes. We boil them in the beaker in the water. Why? But give reason. You are asked this type of a question. Give reason why there is a necessity of boiling that in the water to kill the cells. But why kill the cells? So that in the process or during our experiment, the process of photosynthesis should not happen. During the process, during our experiment, photosynthesis नहीं होना चाहिए. वो तो नॉनसेंस से दिखाएगा. अगर मेरा नॉनसेंस सुनोगे तो एटलीस्ट शायद 5 मार्क एक्स्ट्रा मिलेगा. इसका नॉनसेंस सुनोगे तो तुम्हारा वेस्ट ऑफ टाइम ही है.
डायग्राम रख के
हाँ
आपने डायग्राम रख करके इसका डायग्राम
तुम अपना भोला चेहरा लेके बदमाशी करते रहो.
ना
Boil the leaf in water. So remember the reason. Now the next step is what? Rinse in cold water. Rinse in no. Boil the leaf in water, then boil in alcohol. Boil the leaf in alcohol. That's the next step. Boil the leaf in alcohol. Why? Methylated spirit. Methylated spirit, 70% alcohol, whatever you call it. So now I'm saying boil the leaf in alcohol. क्यों करेंगे boil the leaf in alcohol? To remove all the chlorophyll. To remove all the chlorophyll. We are going to experiment with the leaves, but we don't want them to be green because if they are green, we are not going to understand any color later. So हमें वो जो green leaf जिसपे हमें experiment करना है, but we want them something to be pale now. We want them to be something light green now, some pale color they should develop. Then only we can see the new color developed in them due to the during the course of the experiment. So boil the leaf in alcohol. Remember the reason. Now we rinse in water. Rinse in cold water. After that, what do we do? Apply iodine drops. Apply iodine drops. So I took a leaf something this way, and then I had gone through all these steps now. And after that, when I put some iodine drops on this leaf, what am I going to come across is this particular leaf may show certain regions one particular color and certain regions another particular color. I may come across that certain regions are blue-black, and I may come across certain regions are brown. After this, experiment. Now, when I talk about regions which are blue-black, what does this indicate? Regions which are blue-black indicates the presence of starch. Regions which are brown indicates the absence of starch. So ये हो गया basic experiment जिसपे बाकी सारे experiments are based on. ठीक है? Rinse in cold water only to wash away the alcohol, so that now when you apply iodine drops, alcohol generally allows to dry up any type of a surface. No, alcohol is basically used to evaporate and help in the process of drying up much faster. So अभी अगर हम, if you're not rinsing out the alcohol, the moment you apply the iodine drops all will be removed out much faster. We want the iodine drops to be on the surface of the leaf at least for a few seconds, and then shake it off, allow the leaf to be away from the alcohol, and then away from the iodine, and you observe the color. Now the question is, what is the color of iodine solution? What is the color of iodine solution? Any idea?
Blackish blue.
No. The color of iodine solution is something like
Black.
yellowish orange. It is orangish, almost yellowish orange. So if your teacher shows you in the school, you'll get to see. When we are talking, school में black है, तुम्हारा school में iodine solution black है. Iodine solution black नहीं होता. When we are talking about, हाँ बोलो. That is only about the reaction. Say suppose I take a test tube, and this test tube may I have taken. I'm just giving you out of this experiment, not exactly this. I'm just telling you a simple experiment, which is a very, very prompt experiment to understand the concept of starch and the iodine solution. So I'm not talking about the iodine solution, which is something like yellowish orange, and then you take the starch. सबसे readily starch हम कैसे बना सकते हैं? Take some amount of water and put some flour in it. Flour. हाँ, वो potato starch भी हम डाल सकते हैं, या तो some आटा है, मैदा है, etc. Those wheat flours we can, and then you mix it properly, and then just put one drop of iodine solution. Immediately you will come across the entire thing is becoming, the entire suspension is becoming blue-black. The solution is not blue-black. Remember, the indicator is not blue-black in color. It has got a completely different color. So now, what are we understanding? That we have first of all removed the chlorophyll. But did we remove the starch that was stored in the leaf? No. So what did we do? We removed the chlorophyll. हमने remove क्यों किया? So that now we want the leaf to be a little bit paler in color. Green exactly नहीं चाहिए. अगर green होगा तो हमको ये difference in the color नहीं समझ में आएगा. So we made the leaf a little bit pale. After that, we applied drops of iodine. Now that we put some drops of iodine, what do we get to see? A leaf which has a higher content of starch in it will have major areas blue-black. और जो सारे areas में, जो सारे cells में इतना adequate amount of starch नहीं है, that will appear brown. आया समझ में? This much is clear. Now let us start with the experiments one by one. So this is what I was showing you, how exactly the steps are. Sometimes in your paper, you get a series of these diagrams and then certain questions based on that. Now, let us try understanding the first experiment. Let us start understanding the first experiment. What is done here? The first experiment is we are trying to understand carbon dioxide is necessary. You can see this. Carbon dioxide is necessary for photosynthesis. What has been done here? Try to understand the steps. We are taking a conical flask. Check the similar diagram that we have in your book also. The conical flask. We take a conical flask. And we are selecting certain leaves. We are selecting certain leaves. Here, if you see, they have taken two leaves here. One leaf, completely put it inside the conical flask and close it, cork it. And the other leaf completely outside. या तो कभी-कभार हम same leaf के साथ भी कर सकते हैं, half of it inside and half of it outside. That also can be done. So remember, you may get diagrams in two, three different ways. So either I take one leaf inside, one leaf outside, or I take the same leaf, half of it inside and half of it outside. It's the same thing. Again, something more that we have to take in the conical flask is a chemical compound known as potassium hydroxide. Potassium hydroxide is a chemical compound which readily reacts with carbon dioxide. Remember, it is a compound which starts reaction with carbon dioxide very fast. It can absorb. So now this is the setup. Where should the setup be kept? Under sunlight, normal temperature. Okay. Now, after keeping it for a particular period of time, what do? We remove the core and take out this leaf. So now we have got two leaves. Leaf number 1 and leaf number 2. Leaf number 1 was inside the conical flask for a large for a longer period of time, maybe now for 6 hours. Was inside the conical flask for almost about 4, 5 hours, 6 hours. And this was straight in contact with the sunlight, carbon dioxide, all the parameters necessary for photosynthesis. Now, when we do starch test with both of these leaves, we are now doing starch test with both of these leaves. Now tell me, which leaf is supposed to show blue-black color?
The one outside.
The one outside. क्यों तन्मय?
क्योंकि कार्बन डाइऑक्साइड मिलता है,
इसे कार्बन डाइऑक्साइड भी मिला, टेम्परेचर भी मिला, सनलाइट भी मिला, all the factors necessary to carry out photosynthesis. So it must have prepared glucose and stored adequate amount of starch. So why did not this leaf? Why it could not? This particular leaf got sunlight also, temperature also, because glass will allow all the heat and everything to transfer. But it was not having adequate amount of carbon dioxide because potassium hydroxide was absorbing the carbon dioxide, whatever was trapped inside the conical flask. So the rate of photosynthesis here was much lesser. Glucose formed was much lesser. And hence, starch stored was much lesser. So this leaf will show brown color comparatively.
Very dark. Very dark.
For a longer time, yes. For a longer time. So this is the experiment. So what are we learning from this experiment? What does this prove? It proves that yes, carbon dioxide is very much necessary for the process of photosynthesis, for the manufacturing of glucose and for the storing of starch also. Aaya samajh mein? We move on to the next. Doubts hai pucho. If anything is not understood, please ask. We come to the next one. Sunlight is necessary for the process of photosynthesis. What are we doing? We take some leaves. We select some leaves. Then what do? We cover one region of the leaf. Any region. Now they have covered the middle region of the leaf with some black strips of paper. Pin, both the sides, upper and down surface also. Clip this black strip of paper on the upper as well as on the lower surface. But this leaf, we are not supposed to pluck it out from the stem or from the plant. Let it be there attached with the stem, attached with the potted plant, whichever you select. Let it be there for at least some few hours. Let it be there let it be there for some few hours, at least about 6 to 8 hours. After that, remove this black strips and this leaf go for starch test. So something around after 4 or 5 hours, take out the black strips and ये जो green leaf है, इसके साथ हम ये सारे steps follow करेंगे. So now, what do you expect? What are the steps? First is boil in water, boil in alcohol, rinse in cold water, apply iodine drops. So ये करने के बाद we are going to see that ये जो areas which were not covered, exposed जो areas था, these areas are showing a blue-black color. And जो areas covered था, that is showing a paler or a brownish color. Why? Why, Prisha? This was not receiving sunlight. So sunlight, if it is not adequate enough, chlorophyll activation and the rest of the steps, nothing could happen. The process of photosynthesis was almost negligible. So the mesophyll cells here, could they prepare glucose and starch? Could they store starch? Nothing. So that's the reason a brownish or a pale yellow color, and this one blue black, indicating an adequate amount of glucose and starch present. Sunlight necessary for the process of photosynthesis. Here we talk about the third experiment, chlorophyll. We are always understood chlorophyll is a major component necessary for the process of photosynthesis. Now let us start understanding chlorophyll. Now इसमें experiment में थोड़ा सा difference ये है कि whichever leaf you select, suppose now I select this particular leaf. This leaf has to be a variegated leaf. Variegated. You know what is a variegated leaf? Variegated leaf कौन सा पता है क्या? और leaf which is, say suppose I give you two examples, like mango leaf है और एक money plant leaf है. Difference है दोनों में? क्या difference है? Shape. वाह. किसने बोला ये? इसने बोला. इसने बोला. श्रीराम ने बोला. अरे श्रीराम तुमसे छोटा है. मुझे लग रहा है कि I'm teaching the second standard. Which one is bigger, children? हाँ, mango leaf is bigger, ma'am. हाँ. Money plant has uneven distribution of chloroplast. इसलिए कहीं पे dark green, कहीं पे yellowish. हाँ, whitish, pale yellow होता है. कभी pale yellow है, कभी dark green. अरे now what? तुम उसके पीछे क्यों पड़े हो? Now what? हाँ?
सब लोग उसके पीछे.
सबको तुम लोग eraser, your parents are not giving you erasers. I really want to meet your parents. They must be giving you enough stationery. कहाँ पे तुम लोग कहाँ रख के आते हो?
Pocket में रखे.
What excuse you are giving, asking erasers? अच्छा फटाफट. Now when we are talking about, I said the mango leaf, if you notice, it is evenly green.
Yes.
Meaning there is even distribution of chloroplast. But if I talk about the money plant, as I said, the money plant leaves are variegated, uneven distribution of chloroplast है. कहीं पे ज़्यादा, कहीं पे कम. That's the reason some regions are green and some regions are pale yellow, a light greenish. So that type of a leaf only now we need for this experiment. That type of a leaf, a variegated leaf, हम लेके, and then after that we are going to mark certain areas in that variegated leaf. We are going to mark certain areas. Say suppose now I understand that these are the dark green regions. These are the dark green regions, and maybe these are the light green regions. So I have just marked roughly. एक दो तीन region को ऐसा mark कर लेना है. So we have to just roughly make a sketch of that leaf, and then you go for all the four steps. So ये जो variegated leaf है, इसका first of all we'll take a trace. एक roughly दो तीन जगह which are the dark green, which are the light green areas हम पहले mark करेंगे, and then we'll go through go for all these four steps. Boil in water, boil in.
Leave.
Why do you leave?
Leave को boil करेंगे.
हाँ, leaf को क्या करेंगे? What are the four steps?
हम boil करेंगे.
And then which is that?
Leave करेंगे.
What are the four steps?
हम पहले boil करेंगे.
In
Water.
C's condition. Number two,
Alcohol.
alcohol. Now three, वो देखिए तो बता रहे हैं. I'm not able to cover all that. हाँ, देखिए तो बता रहे हैं.
दिख नहीं रहा. इससे क्या होगा?
Third,
Cold water.
हाँ, fourth, condition is specific. अभी ICU में डालने वाले तुमको. हाँ, फिर hot water दिया है. That also does not matter, but we have to rinse it. Generally, जब हम lab में करते हैं, हम generally we apply cold water. Okay. Now what are we doing here? They इसको भी ICU में डालना पड़ेगा. Next phase. So अभी we have the trace, a rough drawing of the leaf, and we have gone through all the four steps with this leaf. Now after putting drops of iodine, what do you expect? What type of a color will this region give? or after putting iodine, now we don't, in the real leaf, we don't have these markings, it's not so. So in the real leaf, after going through all these steps, we come across that yes, this particular leaf generally shows some regions are blue-black and some regions are brown. Now you can match with the trace, you will come across, the regions which are blue-black are actually the regions which you have marked as dark green. And the regions which are brown are actually those regions which actually you have traced out as light green regions. Why but Aishwarya? Because these were the areas जहाँ पे chloroplast ज़्यादा था, starch accumulation ज़्यादा था, so that's why blue black. And these were the areas where chloroplasts were much lesser and accumulation of starch comparatively much lesser. So what are we understanding from this experiment? We are understanding that chlorophyll is necessary for the process of photosynthesis. We come to the next one. Oxygen is released during photosynthesis. Oxygen is released during photosynthesis. So what are we going to do? First of all, we are going to take a beaker. It is also known as a trough. It is also known as a trough. What is a trough? A big beaker. A trough. So what do? We take a beaker, and now the beaker has to be almost 3/4 filled with water. The beaker has to be almost 3/4 filled with water. Then what do we take? We take a glass funnel. Glass funnel dekha in the lab mein. We take a glass funnel, and in the neck portion of the glass funnel, meaning if I talk about the glass funnel, this funnel now that you take, in the neck portion of the glass funnel, you have to fit in some leaves, some aquatic plants you try to keep inside. You try to fit in some aquatic plants like this. So if you put in some bundle of some aquatic plants, they should be almost tightly fit in the neck of the funnel. Ab jab ye ho gaya, you put it upside down. You place it upside down like this. You are placing it upside down. So already there are some aquatic plants, quite a number of them. We generally put it exactly till the neck region. And now cover the neck region with a test tube. So iske ye jo arrangement of the test tube, that's also something very cautiously you have to do. First of all, we will take a test tube like this, almost 3/4 filled with water. Almost 3/4 filled with water. But ye jo test tube hai, we hold it this way, but we have to directly plunge it upside down just in a few or a couple of seconds. Isse zyada nahi, fatafat we'll have to do it. So it is something like the arrangement of the experiment is very important here. So when I'm saying that ye jo funnel hai and the aquatic plants are all here we have placed, and now the placement of the test tube is very important. Ye jo test tube that we are going to place it here, putting it upside down just in a couple of seconds should be very accurate, otherwise all the water will run out from here. Risha, better concentrate. So the water should not run out from here, it should just get stuck on the surface of that funnel. So the glass funnel is there, the stem portion of the funnel is there. So I take the test tube and just plunge it over the stem portion so that the test tube should get fit in the neck portion of the funnel. It should not be loosely fit also. We have to take it we have to measure the correct size of the test tube also. Otherwise, what will happen is the water will all run out. The water will all run out. The water will all run out. So ye ho gaya arrangement of the apparatus. Then what do? We keep it under sunlight for a particular period of time. Minimum for a period of 4 to 6 hours. Then what do we get to observe? We see that some bubbles start appearing. Under all normal temperatures, whenever we are keeping this, some bubbles start appearing. So ye jo space tha, when we had just started with the experiment, this space was very little. Ye jo space tha, the space was very little when you started with the experiment. but gradually in a period of some hours you get to see that this space is increasing. ये space बढ़ता जा रहा है and these bubbles are continuously rising up, rising up, rising up. Now, what do you want to do is, we want to understand what is this gas getting accumulated here? Which gas is this? Is it carbon dioxide or it is oxygen? Now, we are understanding, okay, we assume that it is oxygen, but we'll have to test it out. Now, in order to test it out, again, the second step is very, very important. Now, the test tube, which was something like this, you have to hold it and take it again just in a second. So that ये जो gas है, by the downward displacement of water, it will just come up and immediately you have to put a burning splinter near the mouth of the test tube. तो अगर आराम से करोगे तुम लोग जैसे करते हो, आराम से ऐसे जब तक करोगे, जब तक ऐसे सीधा करोगे, all the oxygen has come to the atmosphere, और जब तक matchstick लाओगे, nothing is left there. So it has to be done very fast, and you take it upside down, immediately put the matchstick here, burning splinter जिसे बोलते हैं, take a matchstick here, and you will get to see that the fire is even more. The fire is even more. Now, if the fire is even more, then what are we to conclude? Oxygen, because oxygen supports combustion, oxygen supports burning. अगर carbon dioxide होगा तो ये extinguish हो जाएगा. So, what does this experiment prove? Oxygen is released during the process of photosynthesis. Now, ये जो carbon cycle है, that is not there in the syllabus. Carbon cycle is not there in the syllabus, and importance of photosynthesis, already we have discussed about this, about the equation when we have talked about the release of oxygen. These images go through and take these questions as homework, which you will complete. Doubts है next class में पूछोगे. Complete this question. See sub number 1
अरे कृषि वाले ने देखा नॉवल सेक्शन में ये आवाज मत
Blank करना है.
अच्छा, C sub number 1 फटाफट complete करो. C sub number 1 ठीक करो. All these I'm going to check next class when you come. C sub number 1 complete करो. 4 question number 4. 79 का question number 4, number 6, number 9, number 9. Okay, and 3 and question number 3 from 79 and 2 from 89. Circle करके रखो. We are going to discuss it next class. Only these 2 questions I'll discuss in the class. Okay, and this rest of these 3 questions complete करोगे सब लोग.
Yes. ये ऐसा नहीं चलता, ऐसा नहीं होता. मत जा यार. हम तेरी जरूरत है.
मत यार प्लीज यार.
अरे मॉनिटर तो ले ले ना.
कौन सा मॉनिटर?
अरे उधर कर दे पूरा.
उस साइड.
अबे यार मत जा यार. अबे आंदर के मीटिंग है.